We're good to go.
Yeah.
All right. Good morning, folks. My name is Dan Reynolds. I am a first year's master's student here in the honeybee lab at WSU. And I'm going to be talking a little bit about my research. I have not actually started it. Everything's kind of getting in place yet. So, what I'm looking at is evaluating the effects of dietary inputs to enhance Apis mellifera immune response. And this should work. So before that, a little bit about my back history here. How did I get here? Well, I'm originally from Maryland. And if you look at the arrow there and zoom all the way in, you can actually see the home that I grew up in. Not really. And then flash forward, 2015, I got a job at University of Maryland working for Dr. Van Ingelsdorf. And I also worked for the Be Informed Partnership, the nonprofit that is run out of that lab. And that's where I met my wife, Kelly. She was getting her PhD there. She was postdocing for Brandon Hopkins, Dr. Hopkins at WSU. And so I came along for the ride, applied for grad school, and now I'm a master's student. So that's that. Getting right into it. So it's not easy being a bee, as I'm sure most of you know. There's a lot of different factors that are negatively impacting the honeybee. You have pathogens, pesticides, poor nutrition, and parasites. And I just like alliteration also, so that's fun. But today I'm just going to be looking kind of at the nutrition, dietary inputs. So my question is, can dietary inputs increase an immune response in adult honeybees? So in order to do that, you have to figure out how to assess an immune response. So I'm looking at different dietary inputs and then looking to measure immune response on the output of that there. So in order to do that, I will be looking at hemolymph and hemocytes. And hemolymph is basically the blood equivalent for insects. Within the hemolymph, there are these cells that circulate throughout the bee's body called hemocytes, and they act as part of their innate immune system. So when there is some sort of foreign invader that gets into the bee's body, these hemocytes will respond to that. In particular, the plasmatocytes will detect it, and they actually sacrifice themselves as a cell, and they actually surround it and encapsulate this foreign body, preventing it from being able to interact with the host's environment. Then the secondary part is another type of hemocyte, the granulocyte, will release a phenol oxidase. The phenol oxidase will melanize the foreign body, and then this melanin will block the pathogen's function. And basically, the melanin is the same melanin that you would find in your skin. And then so this part of this reaction is it causes it to get darker in color. So in order to extract the hemolymph, what I have to do is a bit morbid, but I do make sure that the bees are dead before I do this. But you remove part of the antenna, and then by pressing down on the thorax, I can get a little bead of hemolymph to come out, and then I pipette it up, and then what I'm looking to do is then place it into one of these wells here on this 96 well plate. The nice thing about the hemolymph is that it doesn't know that if it's inside the bee's body or not. So when you place it in the plate, the hemocytes respond like it's a foreign invader. So they will actually melanize inside one of these little wells. And as you can see, you can't really get very much hemolymph from each bee. This paper that I found this protocol from, they're able to get about 2 microliters, and 1 microliter is 1/1000th of a liter. So it's a very small quantity, and some bees don't even give me 2 microliters. But over here you can see this is hemolymph from 20 bees, and that took a significant amount of time to get. So still working through some of these processes. But here we go. This is what the hemocytes look like at 1,000 magnification. This is without the anticoagulant. and you can kind of see it is also stained, but you can see how the plasmatocytes have spread out, and you can't actually see very many cells left in here because it kind of all glommed together. And then over here is without the anticoagulant, so you can kind of see some of these hemocyte cells and the nucleus inside of them. So back to the inputs, food inputs, honeybees, obviously get a lot of their nutrition from their honey and pollen, but we were just kind of thinking about all the different chemical compounds that you would find in honey and which one of those might be beneficial to give the bees. And when you think about the current state of bees and, during pollination events and that sort of thing, they have, minimal forage, or they don't have as much diversity of forage to choose from. So we were, thinking that maybe by selecting a few of these based on how easy they are to get a hold of and also affordability, we selected these particular compounds. So that's looks like that's where I'm at right now. I hopefully in a couple of weeks we'll have a cage experiment up and running that I can get some more results, and hopefully next time see you folks I'll have more to present on. But looking to run 120 cages, 3 weeks duration, do that 3 times. And then these different dietary inputs, obviously we're going to give it sugar syrup, kind of a good diet of honey and pollen, the polyphenolic compounds, which I just mentioned there, tyrosine, and the fungal extracts that Dr. Nager is working on. We're going to feed there and see what happens. So they could also change, in the future. So as it is right now, that's what I've got. Hopefully, next time I have a couple more minutes, I can add to my presentation. But yeah, thank you for listening.
Okay, thanks. Again, we're going to hold questions till the end. So if you have a question, just remember it for later. We're going to bring up Adam Ware next. Share screen. This one. Okay. Should be good.
So hi, my name is Adam Ware. I'm in Dr. Shepard's lab. A little bit about me is I've been a commercial beekeeper for 5 years. I worked for Kona Queens, Big Island Queens, and I manage Rare Hawaiian honey and done some pollination in California. And the project I'm working on with Dr. Shepard involves a honeybee that's native to the Chinshan Mountains in Central Asia. This region is where the main cultivars of domesticated apples originated from, and so which makes it a particularly interesting honeybee for apple pollination, as it co-evolved alongside apples in this region. So this is a particularly beautiful picture. This is one of the sites that Dr. Shepard collected some of the drone semen for insemination.
Sometimes you have to change it first on the laptop and then.
All right. Yeah, so this is Chinshan mountain range, and these are some of the areas where Shepard collected the Apis pulmonella, and this is the native apple tree that comes from that region. So my project is really helping Dr. Shepard sort of bring out this bee out of the cryopreservation tank, where looking to sort of assess its potential for breeding and apple pollination, to incorporate also into maybe our breeding program. This is what it looks like. It's kind of similar to the bees that we have here. It's related, it's similar to the Caucasian bee. And so this is sort of important because introducing diversity is beneficial for your breeding program. And it minimizes the likelihood of genetic bottlenecks when we're selecting for desirable traits. You know where this is at?
Yeah, that's where Brandon and I collected, that's right near the border with China.
This is Brandon. He's collecting drone semen. I guess, I don't know, I kind of want to play this. If you've never collected drones, this is what we collect the drones out of the hive with. And it's kind of easier, and then we'll stick them in this sort of cube, sort of cage, and it just makes it easier extracting. And so the rationale behind this is that we think that this particular honeybee is advantageous to the Pacific Northwest. It could be a good overwintering bee. It's well adapted to cold weather, potentially, you think. And it could, we could potentially get some novel characteristics for breeding that we can incorporate into our breeding program. Ooh, so this is Sue Kobe. She inseminated all of our. So the way we learn about it is they get Caucasian versions, and we inseminated them, frozen drone Pomonella semen. And this is the first one that we got. And I kind of wanted to show you this because you can kind of see that the pattern sort of, it's a little droney. So sometimes this semen came from 2015, so it's 7 years old. So sometimes we'll get a really nice pattern, sometimes not. And so you're kind of just grafting in the dark, it's just whatever you can get. You don't really know if it's going to be drone or worker. So that's kind of what I've been doing. I've just been grafting like crazy, trying to get as many as I can out of it. So yeah, I kind of summarized this already. Soon, inseminating their queens. So really for this next year, what I'm trying to do is overwinter the queens that we got, and then repeat this as many times as possible to get to the highest percentage toward Pomonella that we can get. Usually the most you can get is about 97%. That's all I got. I guess we'll do that.
Yeah, we'll do questions at the end. And we're gonna bring up Riley Reed next.
Hey, Riley Reed.
Sometimes you have to change the first slide on the computer, and then it will like change it quicker. And now it might work, and then the green one's timer. Okay.
Yes, I'm Riley. I'm Brandon Hopkins, PhD student. I actually grew up about 20 minutes from here, and now I went over to Pullman, got a job working there, and decided to stick around for my PhD in Brandon's lab. Today we're talking about my project, or one of my projects, which is improving crop isolation through supplemental feeding. It's particularly focused on stuff like carrot seed and onion seed. So yeah, a little bit about the problem. When you get these seed crops, you really want to keep the seed purity high. If you have, like, if you're growing one variety of carrot seed and your neighbor's growing another variety, and the bees are flying back and forth and spreading the pollen back and forth, your seed's not going to be worth anything because you're not really going to have either variety. You're just going to have a mixture. And that's particularly important since honeybees, that's what we normally use for pollinating just about everything. But they're really good at foraging long distances when they need to. And then they'll bring some of that pollen from your field to your neighbors, or your bees may bring pollen from your neighbor's field back to yours, and it's kind of back and forth. And this becomes a bigger issue with monoculture, especially with some of the crops I don't really like carrot seed. Where it's really not meant to be pollinated by honeybees, it's meant to attract stuff like flies. So the honeybees really don't like it, and they're trying to go out and fulfill like different nutrient deficiencies by visiting other plants. It's been shown they'll ignore forage and find other further forage if they are trying to meet these needs, like missing amino acids and stuff. Yeah, and we always hear bees will forage up to 3 miles, but there's been some studies showing when they are trying to meet those nutritional needs, they'll forage potentially up to 6 miles. Which means if we have hives sitting outside of this building and they can't find good stuff around here, they may be in like the garden center at Walmart in Othello. So that's pretty big problem, especially since currently isolation distances for a lot of these crops are not that far, they're more like a couple miles. So kind of the solution we're proposing, based off of this knowledge of these trying to go out and fill those nutrient deficiencies, is supplemental forage and in-hive feeding right now. Eventually we'll try to add in, if this works, we'll try to add in supplemental forage around the fields. This is much easier to implement in the short term than trying to figure out what plants we can plant around the field without messing up the grower. So we're doing these pollen patties and sugar syrup, those specific pollen patties and sugar syrup. This will remove their need to fly quite as far. If we can meet their needs there, they don't have to go out miles away to find forage. And there's been some research kind of like this done with kiwis, which have a similar issue of the bees don't like foraging on the kiwis. I'm not 100% sure why, but it sounds like it's similar to the carrots, just they don't attract bees very well. And they found when they fed sugar syrup, and pollen patties both, that the bees would quit flying as far. They bring in less pollen from other plants and more from the kiwis, since kiwi growth was right around the hive. So it seems like when they have these nutritional needs, they don't quit foraging, they forage around the hive more than they normally would. So this will improve the amount of pollination on the carrot seed and decrease how much they're bringing in. Oh yeah, that'll result in decreased pollen transfer between fields. So we're gonna really start next year. This year's just kind of been me troubleshooting this, because in order to track where the bees are going, we want to be able to mark them. And bees are very good at cleaning themselves. So we started out, there's been some other researchers that have had good luck with these colored powders. They're like what's used for like gender reveal parties and stuff like that. So you get like pink and blue powders and stuff like that. And there's a few different methods. One of them is this, like you put a broom on the front coated in it. You can put little like bags hanging over the entrance and a few other options to try to coat the bees. You can see it coats them pretty well, but they clean it off. I have little pouches I put on front of the hives with this powder. They'd have to brush up against to coat them. And it would work at first for about 15 minutes, and then they figure out how to clean it up. And after that, it quickly got to where it took like 2 minutes, and just all the powder would be gone. So then we tried hand painting, kind of like we do with queens. But that's very time consuming. And when you have something like some of these big fields and stuff with hundreds of hives around them, we painted like 2,000 bees. That's not enough to go out and find them in the field. So we scaled up a spray paint. The bees survived. We got these spray paints like what they used to grass and stuff, and it didn't seem to hurt the bees. Once we got paint, we killed a few and we really covered them. But surprisingly, they cleaned it all off. Like we spray painted 4 or 5 frames in every hive. Came back a few days later, and there was like 5 bees in each hive I painted. And there weren't a bunch of like dead painted ones I could find anywhere outside the hive, or I walked out and field some of this. And just all cleaned it off. The frames not so much. All of our frames are really colorful. So I'd go a little bit more destructive. In order to do it now, what we're going to try is food coloring. We found if we mix in food coloring into the sugar syrup, and then for the untreated colonies, we'll try mixing it with water. Surprisingly, the foragers are leaving the hives with their guts full of syrup. But I didn't fully expect this to work, because I thought they'd be hungry, but they leave on a full stomach. And you can see right there, it stands as really bright blue. It's some of them, even before you get them open, they just look kind of blue. So that's what we're going to be, or that's what I'm going to be working on next year. This just kind of shows what the fields will look like. So we'll have like a center pivot, or maybe it'll be a different shaped field. And in each corner, we'll have 2 different groups of hives. One group will be fed sugar syrup pollen patties, and the other group won't be fed anything. And then, well, they'll be fed water with the food dye, so we can still get them colored. And then I'll go out around the field and do sweep netting and try to collect a bunch of foragers in the field. And compare the number of each color of bee that we find in the field to how many bees are actually leaving each of the colonies. So we can kind of compare. One has like, if one group has twice as many foragers going out, but half as many in the field, we can assume that there's a lot of foragers going somewhere other than the field. Yeah, kind of expected outcomes from this. get a new method of decreasing the pollen transfer between fields and be able to come up with some updated recommendations for isolation distances for the growers. And this could mean fields are closer together, and if they can put fields closer together, they can have more fields of, like, say, carrot seed in an area, and hopefully it'll increase the number of pollination contracts for those seeds or for those seed crops. And then I've got a few other kind of smaller projects. The other we're kind of working on kind of along the same line of improving pollination on fields is whether or not hungry hives are better than like heavy, full, strong hives. There's been some studies showing that when foragers get back to the hive, if there's no space for them to put nectar, there's no bees to take that nectar from them and put it away, they're not going to forage as much. So it may be that you actually want smaller, weaker, like or hives have been like sitting somewhere they haven't been getting a lot of food to go onto a field, so they're really eager to go out and forage. I'm missing a word there. That doesn't specifically have to improve immune function. So what the second one is supposed to say? There's that Vitahealth company that you may have heard Brandon talk about yesterday that's trying to get the glycerin and oxalic acid formulations registered. They're also testing out artistic acid feeding for bees. It's been shown in kind of a few different animals to improve immune function. Hopefully that'll fight off some diseases and stuff. And then what this picture is kind of relevant to is ozone sterilization of frames. It's not been tested much on the commercial scale, but in labs, it's been shown that with small samples, you can kill wax moths, foul brood, and you can break down pesticides on these frames. It's just kind of a cool picture. You can actually diagnose AFB with the scales that glow under a black light. So you guys buy a UV black light online and get a pretty good idea what you got going on in your hive. Thanks.
Thanks, Riley. All right, and then last but not least, we have Kirsten Ritchie. And then we have this one. Okay.
All right. All right. Hi, everyone. My name is Kirsten Ritchie. I'm a PhD candidate in Dr. Tim Shepard's lab at the Department of Ecology. And I'll be talking to you about my project involving the reproductive mechanism of evolutionary differentiation in the honeybee. So honeybees are thought to have originated in Asia. Their native ranges are within Europe, Africa, and of course Asia. They made it to the Americas by human migration, and that's why we have our lovely pollinators here with us. They are separated into 28 distinct subspecies, and those subspecies are then further categorized into what we call evolutionary lineages that were defined by an Austrian beekeeper called Friedrich Rutner. He did a lot of morphometric and geographic analysis to categorize these honeybees. And so you can see. So in this image, you can see this gradient of kind of genetic relatedness among the honeybee subspecies. And these are kind of the four main ones. So A from Africa, O from Asia, M from Western Europe, and then the C lineage from the Northern Mediterranean. And so all these subspecies are well adapted to different environmental factors because they're based in all of these different locations. So these images just show the vast morphologies and ecologies where these honeybees exist. So you can see hopefully these very yellow bees and kind of darker bees, and then they live in these really hot, dry environments or kind of colder, arid environments. And so my work will involve the use of different US strains, as they're called. They aren't actual subspecies from the old world, but they represent these evolutionary lineages that I mentioned before. And I will be using these strains to look at differentials for Muse by queens So here are just some examples of strains that I'll be using: Italians, Africanized bees, and Carniolan and Caucasian bees. So queens, honeybees have this really cool way of reproducing. As we all know, they have a single reproductive female in the colony, the queen. And early in her life, she will go on up to 4 mating flights, where she mates with up to 20 drones in flight. After copulation, there's a time difference between the mating, storing of the sperm, and then actual fertilization of the eggs. So in this image here in the vaginal opening is where the drones will deliver their semen, where it all gets mixed together from multiple drones in the common oviduct, where afterwards it either gets expelled back out the vaginal opening or makes its way to the spermathecal duct and migrates to the spermatheca, or the long-term sperm storage organ. And once there, the queen will use that sperm for the rest of her life. She won't go on any more mating flights. What she has is what she gets. And so it's really important, the sperm that she collects. So again, she doesn't use all the sperm that she receives. Sometimes she'll eject it. And then even once it's stored, she might not use all that sperm in her lifetime. So I'm going to be looking at kind of the female-male interactions within the reproductive tract to see if there is differential or preferential storage of the sperm from different strains that it mates with, and then even further, if there's preference for what sperm she actually uses to fertilize eggs. And so this is a really cool project because it will hopefully help us glean insight into mechanisms for asymmetric gene flow. We've seen this in between African and European honeybees, where in these hybrid zones, the progeny from European and African crosses show they look and behave more Africanized, and there's also molecular evidence to show that there are a higher percentage of African genes in these populations. So some suggested mechanisms for this is a higher colony growth or swarming rate of Africanized bees, so that there are just more numbers of them in an area. They will swarm up to like 6-12 times a year compared to European honeybees that swarm maybe twice a year. There could be advantages for queens sired by African drones. They have shorter development time so that they can emerge sooner and kill all the other emerging queens. Some have suggested there is dominance of African alleles over European alleles, but that is very highly debated. There could be genetic incompatibilities between European maternal and African paternal alleles, whereby process of this nature with the European queen and African drones just don't produce viable progeny. And then there could also be this differential sperm use, which is what my project will kind of look more into. So a study done in 2003 showed that when European and African queens were inseminated with both with semen from both European and African drones, on average, there was a higher percentage of workers with African paternity. And so my work will follow this kind of same line of questioning, but carried out longer than this, hopefully longer than the 6 months that the study was carried out, and also look at not just progeny, but spermatical contents and relative sperm storage. So here are my research questions. I want to know if there's a difference in the number of sperm that the queen stores when she's inseminated with equal amounts of volume, equal volumes of semen from drones of either the same and or different strains. And then I also want to explore if there's a difference in sperm used by honeybee queens. Again, when they're inseminated with equal volumes of semen from drones of multiple strains. So I've done some preliminary work with Italian and Caucasian honeybees. Those are from different evolutionary lineages of very related genetic relatedness. And this is accomplished by taking the queen, instrumentally inseminating them, and then I have looked at the spermicide contents to see if there's a difference in what the queen is storing. And the preliminary data shows that it does seem like the queen will store more of the right type of sperm compared to the wrong or incorrect type of sperm. But again, that was only done with Italian and Caucasian honeybees. I'll be carrying this out again with other strains, including Africanized honeybees. And then eventually looking at the population genetics to see differences in the paternity of these colonies. So hopefully my research will have, the outcomes will have some far-reaching impacts. I hope to get a better understanding of the honeybee mating biology and understanding the female to male interactions occurring in the reproductive tract, and why we're seeing this difference in gene flow among different populations of bees. And on the more practical side, I'm hoping it might help beekeepers and queen breeders in choosing what queens or what bees to use in crosses to produce a better queen or better bees. So that's the gist of my research. Here are my references. Thank you very much.
I think let's bring all four back up. All right. We have about 10 minutes for questions, so we have plenty of time. And, I also just want to say, like, we're really lucky to have such great and hardworking grad students. They are balancing, doing all the research they talked about today and classes and helping us with hive management and lots of other things we asked them to do, modeling all our auction items last night. So we're very lucky to have them. So yeah, does anyone have any questions for any of our grad students?
So is this all the grad students that you have?
We have one more. We have Leary who just started. And remember, he was our Vanna White last night during the auction. Yes.
So Riley, you're having trouble marking your bees. You can use a fluorescent protein stain dye that the bees have to walk through. It's permanent, they can't pull it off. So if you put a wet pad in front of your beehive, I mean, I've used protein stains for years. They just walk through it. They'll be stained permanently, go out in the field with the UV light. They'll fluoresce in multiple colors, so you can do, there's a whole variety of colors. And it's simple to pick it up, and they can't pull it off because it's a permanent dye.
That sounds pretty much perfect.
And then the other thing is, I don't know, it's, you can talk with Brandon and Steve about this, but you could also do put a sensor with a beam brake and a micro injector on top. So every time a bee comes out, they automatically get sprayed on top. So you could actually mark every bee that wanted to come out. It'd be very simple to set up.
Okay.
Any other questions? Yeah, go ahead.
So Adam, can you describe the process of back reading to get that 97% you're talking about when you're?
Oh, sure, yeah. I mean, so we're starting out with, we're using Caucasian virgins, and so we'll have these Caucasian virgins and they'll simulate Salmonella, and then we'll graft off that, and we'll basically keep doing that until it gets closer to 97. You get like halfway each time, so it takes about 6 times to get to 97%.
Is there some way you can tell what percentage they are? Versus the way you do genetics.
Wait, see that?
Is there a way to tell, like scientifically, how much of the genetics is the new genes versus what was originally? I know you started, so you started with the Caucasian. Can you tell how much Caucasians left genetically somehow?
I guess you could like, I'm not actually sure.
It's a good sequence of genome.
Yeah,
Well, you could get a set of highly polymorphic genetic markers. That, you know, characterize the key starting populations and then measure the. That's a little more complex than what we were gonna do, but you can also mark the queens and make them.
Oh, yeah,
And
yeah.
because you're doing instrumental activation.
You still, you do back us 300 times, you know they.
Right, yeah, so the first ones are 50%. That, like you said, it'd be 75% and 87. And each time you get 20, it's also you keep track of your breeder queens, and you know what they are. What's the problem here, Adam, because of where we live, the problem is these things in the winter.
Yeah, our season's kind of short, so we only get a couple tries each year. So I mean, really, it's kind of challenging to get as many back crops as we can before we get to the winter.
Okay, anybody else? George?
I'm just wondering, since I was thinking of your slide of glaciers, will be the years and so on, and I was just wondering, I'm assuming that the apple tree, because I didn't get pushed down when the glaciers came through, they've been there all along and they didn't walk away. These did, but now the bees are there. And I'm just wondering, is it really true that they grew up together and or did they just happen to be there now?
So, let me understand the question. You're saying the glacier came down, but apple trees continued to live under it. The plants and the animals have the same issue with weight.
So they're both glacially manifestations measuring.
I don't know about apple. The evolutionary history of that.
And there's another question, it's not fair to, I'm not really asking you, but. Have there been any studies to see if those bees actually? I know they are pollinating apples there. But if you had a different honeybee, it would also work on apples, like they do here.
Right.
Is there a difference in their ability to pollinate?
Well, that's what we want to look at. We think that they potentially have adapted, you know, there may be some unforeseen advantage to using pollinating here. But we don't. But we think that there's good potential for overwintering and, you know, some unforeseen behavior since they go evolve with apples in that region. Or maybe they just have some traits and we can kind of sort of select for those and bring them extra.
So one thing, George, is, very little is known about that baby. No one, I mean, I think there's a little bit of Russian literature. And, no, we described it, and as far as I know, no one's really studied it.
I know apple, the Malus cerevisiae is from a Russian botanist. That's where it's the name.
Any other questions?
Yeah, I got a question. I forgot your name. Dan. Dan, yeah. So what was the question behind your research?
Is looking whether or not nutritional or I guess dietary inputs can increase in an immune response and a measurable sort of way.
And you said that you haven't really started yet.
I'm in the preliminary stages right now, so hopefully in the next couple of weeks, it'll.
So I would like to get your contact information. I want to keep in contact with you to see how that's going.
Okay,
I'm interested in that as well.
okay, all right, cool.
I have a question. So I'm interested in the reproductive science. So there, it seems from my layman's standpoint, that there's a couple options, either that to determine why the Africanized are so successful. And it's either they're stronger swimmers or the environment as in the spermatheca is better for them. And I'm wondering if to determine if they're stronger swimmers, do the queens release, it seems like they release a measured amount of sperm rather than a single sperm. And could you then, I don't know if the science is there, if you can then test and see if the Africanized sperm are actually in the same quantity fertilizing more often.
So, there was work done by WASBA a few years ago to show that the queen does release like a set volume from her spermatheca, and that contains like 10, 20-ish sperm each time. And but then as far as the seeing, you know, if the Africanized fertilize more than a different strain, there's definitely ways to do that, but that is right now not within the scope of my research. Again, the Bayer study kind of looked at how much sperm is released from the spermatheca during fertilization, and then like which sperm were fertilizing the eggs. So the methods are out there, it's just not what I'm working on right now. But yeah, no, that's a great line of thought.
Then Riley, how big of an area are you using to conduct your research?
So that is kind of dependent upon the growers and VGO seed. It's depends on how big of their fields are. We've been asking for kind of smaller fields, so it's easier to manage, like randomly surveying different parts of that field then. We may be able to scale it up going forward. But I think last year it was 40 acres, I think roughly. And it got some pretty, like much larger fields that we could potentially work on.
I think we have time for one more question.
Riley, on the ozone thing, is that something we haven't really heard that before? I've heard the irradiating above in Canada.
Yeah, there's been a few like lab-based studies, not doing like with full frames of it, like with like they use like actual scales from foul brood or like chunks of wax or chunks of wood stuff out of a hive. And put them into like little incubators that they can monitor the ozone and everything in. Did it in there, they had really good success in there. They were able to kill the wax moths, kill like AFP spores, even break down some of the pesticides and stuff like that. But there's not really been much to like, can we actually implement this real world? Because it's much easier to do on a really small scale. There's a few different commercial beekeepers are really interested in this, and have like reached out to brand and stuff, and they're already buying the ozone generators to try them out. So I'm able to just kind of come through and while they're already trying out their own stuff, I can like add a few extra samples and like go and see how it's working and stuff like that. But there's not been much to actually test like on a practical scale of it, like it works. Yeah, like what we're looking at is like filling up like the shipping containers out back and stuff, like big containers filled versus like I scraped like an inch of wax off of a frame and stuck it in a little box.
Okay, let's thank our grad students one more time. All done.